Welcome to another episode from the Veterinary Research Channel. Have you ever wondered about the complexities of respiratory conditions in our equine companions? In this episode, we delve into a fascinating piece of research published in the Journal of Veterinary Internal Medicine in 2024. The study, titled Microarray Molecular Mapping of Horses with Severe Asthma, explores the intricate world of severe asthma in horses, a condition that bears striking similarities to human asthma. This debilitating allergic respiratory disease is quite prevalent, affecting a significant portion of horses in the Northern Hemisphere. The hallmark of this condition is an elevated level of specific immunoglobulin E, often abbreviated as IgE, directed against various environmental proteins. The authors of this study, Samuel J. White, Laurent Kudel, Eric A. Richard, Elaine Marty, and Philippe B. Wilson, recognize that while we know severe asthma in horses involves these allergic responses, our understanding of how different environmental exposures influence these specific IgE profiles remains limited. This gap in knowledge is crucial because a better understanding could pave the way for improved diagnostic tools and more effective therapeutic strategies. To address this, the researchers embarked on an ambitious project, analyzing a group of 35 sports horses. These horses were carefully selected from 3 different geographical locations: France, the United States, and Canada. Importantly, within each country, the researchers included horses diagnosed with severe asthma and a control group of healthy horses. This careful matching of horses within the same environment was crucial to account for the potential influence of local surroundings on allergic responses. All the horses within each international group were housed in the same facility and underwent the same management procedures. The study employed a sophisticated technique called serological antigen microarray profiling to investigate the sensitization profiles of both the affected and the healthy horses. This method allowed the researchers to simultaneously screen for the presence of specific IgE antibodies against a vast array of potential allergens. Think of it as a comprehensive allergy test looking at reactions to a wide spectrum of substances present in the horse's environments. The goal was to identify and rank the importance of different allergens in distinguishing between horses with severe asthma and those without. To achieve this, they used a statistical method known as partial least square discriminant analysis, often abbreviated as PLSDA. This powerful analytical tool helped them determine which allergens were most strongly associated with severe asthma in each geographical region. The importance of each allergen was assessed using a measure called variable influence of projection, or VIP. Furthermore, they conducted frequency analysis to identify allergens that were commonly important across all 3 international locations. The researchers emphasized that despite advancements in diagnosing severe asthma based on clinical signs and ancillary tests, commercial application of in vitro allergen assessment has been hindered by the lack of robust statistical methods for clear disease classification and the limited number of allergens typically tested. Their innovative microarray platform aimed to overcome these limitations by providing a more comprehensive profile of allergen-specific IgE. Previous work using similar microarray technology had suggested that horses with severe asthma tend to have a broad sensitization profile, involving allergens like latex, fungi, mites, and pollen, mirroring some of the common allergens found in human allergic asthma. The researchers also noted the relevance of these similarities in the context of One Health, the concept that the health of humans, animals, and the environment are interconnected. Learning more about asthma in horses could potentially offer valuable insights into the human condition and vice versa. However, the study authors highlighted a crucial gap in our understanding: the relationship between geographical location and the results of extensive allergen profiling had not been adequately explored. They also pointed out that previous research indicated that analyzing a geographically mixed group of horses with severe asthma resulted in lower accuracy for disease prediction compared to studying environmentally matched groups. This led to the hypothesis that allergen-specific serum IgE levels could vary significantly between different horse populations and due to various environmental factors. Studies in human twins further support the substantial role of environmental factors in shaping specific serum IgE levels. Therefore, this study was designed to specifically evaluate the impact of geographical location on comprehensive IgE profiling and its effects on sensitization patterns and the diagnostic effectiveness of microarrays in horses with severe equine asthma, or SEA. Now, let's turn our attention to the exciting findings of this research. The results of the partial least square discriminate analysis models demonstrated a significant difference in their ability to predict severe asthma in horses across the 3 geographical regions. The model showed a high discriminatory power in horses from Canada, with an area under the curve, or AUC, of 0.995, and in horses from France, with an AUC of 0.867. The AUC is a measure of how well the model can distinguish between affected and non-affected individuals, with a value closer to 1 indicating better performance. However, in contrast, the model performed poorly in horses from the United States, with a much lower AUC of 0.38. This stark difference highlights the variability in sensitization profiles and the effectiveness of this diagnostic approach depending on the geographical location. Furthermore, the researchers identified the specific allergens that were most important for distinguishing between the severe asthma and control groups within each country. These allergens, identified through the variable influence of projection scores, vary considerably between Canada, France, and the United States. This underscored the idea that local environmental exposures play a crucial role in shaping the specific IgE responses in horses with severe asthma. Interestingly, despite the regional differences, the frequency scoring analysis revealed that only 4 allergens were consistently shared as important for classification across all 3 geographical groups. These shared allergens were HEVB 5.0101, CND, DER-P2, and RUM-CR. These findings, visualized through statistical analyses and receiver operating characteristic curves, clearly demonstrated the varied nature of sensitization profiles across different geographical locations and the corresponding variability in the predictive power of the mathematical models used for disease classification. The receiver operating characteristic curves illustrated the balance between the model's sensitivity, its ability to correctly identify horses with severe asthma, and its specificity, its ability to correctly identify healthy horses. The stark contrast in the AUC values between the regions strongly suggested that the allergen landscape associated with severe asthma differs significantly depending on where the horses reside. Now, let's delve into the discussion of these intriguing results. The researchers concluded that their international study revealed significant regional variations in IgE-sensitization profiles, strongly influenced by the specific environmental factors present in each location. This finding emphasizes the paramount importance of considering local allergen exposure when investigating severe asthma in horses. The fact that the mathematical modeling for disease classification exhibited varying discriminatory power across the regions further underscores the need for region-specific allergen identification to develop accurate diagnostic tools. Despite the regional variations, the identification of common allergens shared across all regions is a significant finding. These shared allergens, HEVB 5.0101, CND, DER-P2, and RUM-CR, hold potential significance in developing precision medicine approaches for the diagnosis and treatment of severe asthma in horses on a more global scale. The study highlights the power of microarray technology in analyzing IgE responses and identifying allergen components linked to asthma. Furthermore, it reinforces the reliability of IgE microarray analysis when compared to traditional laboratory methods. While mathematical modeling of profiling data is a well-established technique in human allergy research, its application in veterinary medicine has been relatively limited. This study successfully utilized advanced technological developments and mathematical modeling to specifically explore the impact of geographical location on severe equine asthma. The variable influence of projection outputs provided clear evidence of the variability in sensitization profiles between the different geographical locations. This variability strongly suggests that the likelihood of a horse becoming sensitized to particular allergens is heavily influenced by local exposure, which is in turn shaped by factors such as climate and vegetation. The differences in AUC values for the predictive models across the regions indicate that a single mathematical model might not be universally effective as a diagnostic tool for severe asthma in horses worldwide. Instead, the study emphasizes the importance of considering individual sensitization profiles on a case-by-case basis for clinical recommendations. The identification of shared allergens through frequency scoring, all of which had been previously implicated in severe equine asthma in geographically mixed groups, offers a promising avenue for future research. The researchers suggest that with broader geographical assessment, it might be possible to establish a core set of globally relevant allergens. This could potentially lead to the development of more robust mathematical models capable of providing a more definitive diagnosis of severe asthma. The contrasting performance of the partial least square discriminate analysis models, with high accuracy in Canada and France, but poor accuracy in the United States, strongly suggests that the specific allergen profiles associated with severe asthma differ significantly between these regions. These differences likely impact the effectiveness of allergen profiling and mathematical modeling for disease classification. The observed regional variations in specific IgE levels align with the understanding that environmental factors play a substantial role in influencing these immune responses in horses. Previous studies have also demonstrated significant variations in specific IgE levels among different stud farms, further supporting the influence of local environmental factors. The researchers also drew parallels with human studies that have validated the existence of country-specific molecular sensitization patterns. In their study, they noted marked discrepancies in temperature between the United States, Canada, and France, suggesting that these climatic differences could contribute to variations in allergen exposure, such as pollen levels. The potentially higher rates of pollen sensitization in the United States might be linked to the influence of elevated temperatures on pollen production. Furthermore, the ubiquitous nature of Group 1 allergens across different grass species could also contribute to a higher overall prevalence of grass pollen sensitization in the United States. The study also observed distinct tree pollen sensitization profiles that clearly reflected the different tree species present in each geographical region. For instance, Cas was prominent in the Canadian group, while Kr was specific to the American group. The presence of dust mite allergens, DER-P and DER-F, which are known to be influenced by humidity, was consistently significant for class separation across all groups. This finding could be related to the relative humidity of the sampling areas, as well as the humidity within the stabling environment, which could be affected by factors like forage and bedding moisture content. In contrast, Alternaria alternata, a fungus known to be an outdoor allergen, thriving in warmer climates, and sometimes found indoors, was not found to be significant for any of the group predictions in this study, despite previous associations with severe equine asthma. The identification of HEVB 5.0101, CND, DER-P2, and RUM-CR as the four variable influence of projection scores shared across all three regions is particularly noteworthy. These are all known allergens associated with respiratory allergies in horses. HEVB 5.0101 is a major latex allergen linked to asthma symptoms in latex-sensitized humans and is a notable allergen in severe equine asthma. CYND is a major allergen from Bermuda grass pollen, implicated in both allergic rhinitis and asthma in humans and horses. DER-P2 is a significant dust mite allergen and a known risk factor for asthma. RUM-CR is a major allergen from common curled dock pollen, a common cause of allergic respiratory issues. The consistent presence of these allergens as important contributors to disease classification across different geographical regions suggests their potential global role in the development of severe equine asthma. The researchers emphasize the utility of frequency scoring analysis in identifying such shared important variables. Interestingly, another previously identified significant allergen for severe equine asthma classification, RASP-F8, was only significant in the Canadian and French groups, further highlighting the complex interplay with local environmental exposures. The study's most significant finding is the detection of diverse molecular IgE-sensitization profiles across different regions globally. This has profound implications for precision medicine approaches, such as targeted allergen avoidance strategies and the precise prescription of allergen-specific immunotherapy. Molecular diagnosis is particularly crucial for accurately identifying the specific allergens causing sensitization, which might be masked by cross-reactivity when using whole allergen extracts. This underscores the necessity for accurate allergen identification and avoidance in personalized treatment plans. Given the increasing recognition of the horse as a valuable model for human asthma, this research also holds substantial relevance within the One Health framework. In conclusion, this study has provided valuable insights into the significant role of environmental factors in equine asthma and underscores the importance of considering these factors in future research on serum IgE levels. The findings suggest that allergen-specific IgE levels are influenced by a range of environmental variables, and further research is needed to fully unravel the intricate relationship between diagnosis, environment, and allergen-specific IgE in horses. Ultimately, the results of this study could contribute to the development of more targeted and effective strategies for diagnosing, treating, and managing equine asthma, leading to improved health outcomes for affected horses. Thank you for listening to the Veterinary Research Channel. A link for the reference paper is in the video description. Remember to test your knowledge by answering the questions in the description, and as always, keep those minds inspired, hearts light, and tails wagging.